Oh, no more people here than I expected. I'm going to go turn my lights on. All right, so kind of because of my screw up over the exam, we just have this session open, right? We don't have any material to cover, right? We've covered all of the material that will be on the exam. And this is really just for you to ask any questions, clarify stuff, figure out what is what, that kind of stuff. So it's pretty much just me sitting here drinking my coffee, answering questions. So if anybody wants to kick things off, they are very welcome to do so. More questions, the better. Or you'll just sit here watching me drink my coffee, which may be interesting. I don't know, but I also have a cereal bar as well, which I was planning on eating if no one showed up. All right. What I could do is I could just go through the participant list and pick a person in turn. Shall we do that? For a question. All right. A bit of wind noise going on here. Okay. Ashley, you got a question? Any question? And do I have a question from Ashley? No? That's a good answer. All right, Bridget.
None specifically. I was just hoping maybe other people would, and you just get to talk about topics in more depth.
Okay. Is there anything that you feel particularly happy about in terms of the material? All right, Adriana's messed up my alphabetical order question asking now. Adriana, do you have any questions?
No.
No? Okay. Jacqueline, so it was really an issue of time, right? So we ran out of time because we spent a lot more time covering control of gene expression than I anticipated. And so the only specifically part of the biotechnology PowerPoint 20, that will be exam worthy material, pardon me, is CRISPR-Cas9. However, we have covered kind of in passing, right, as examples, some examples of kind of transgenic work, such as the green fluorescent protein reporters that I've used, and I've shown you in the what is it, the alligator embryo, stuff like that. So in terms of the actual PowerPoints, only CRISPR-Cas9 from PowerPoint 20. And that's kind of a pretty crazy topic in and of itself. And none of the rest, just because we ran out of time. Good question. Yay, first question of the day. Did that help? You're welcome. Cynthia, I bet you've got questions.
I do.
And fire away. If no one else asks questions, literally, Cynthia, you've got an entire hour and 9 minutes.
Oh goodness. Oh goodness.
Yeah.
I did have. I'm trying to get to a parking space because I'm just getting to school right now.
Oh, I don't want you to like crash in someone's car or get a ticket or anything like that because you're asking questions.
Right. Let me park and then I'll ask a question.
Is this front end in parking or are you reversing in?
It's the parallel parking on campus. It's the ones toward the outside.
Oh, that's challenging. Okay,
It
quiet for a
is,
minute.
and there's people crossing and opening car doors. So yeah, let me just park.
Okay, I absolutely suck at parking, as my kids frequently point out, which I think is a little rude. All right, while Cynthia's parking her car safely and not hitting any pedestrians, does anybody have questions in the meantime?
I have a question.
Go for it.
Okay, so when we were talking about the LAC operon, when there's high CAMP levels, then there's a lot of gene expression, right?
Correct.
And how does that work? What do you mean by high gene expression? Like various rounds of like translation and transcription, or like?
So most prokaryotic gene expression is controlled at the level of transcription.
Mm-hmm.
If a transcript is made, then it will, with some exceptions, be translated, right? So that's kind of one of the first things to stress, I guess, that's different between prokaryotes and eukaryotes. Eukaryotes have multiple levels of control. Prokaryotes are really all about control of transcription. So what we mean by higher levels of expression of the LAC operon when cyclic AMP is high is that the RNA polymerase will initiate transcription more frequently.
Mm-hmm.
So the whole purpose of the catabolite activator protein is to help, I'm not sure if it's recruit or retain RNA polymerase, right? So RNA polymerase is this large complex of proteins that kind of, assembles in particular order on a piece of DNA and then starts, right? And it's not especially good at doing that. I mean, it will do. It's actually better in prokaryotes, I think, than eukaryotes. But it can also fall off, right? So for one thing, it needs to, quite often actually needs to be told, come here, right? More so in eukaryotes than prokaryotes, but also once it's assembled, it needs to be kind of like stopped from falling to pieces. Which seems kind of weird to think about, but it's this large conglomerate of proteins, right? Once it gets going, right, and the RNA polymerase itself kind of gets off to the races, it's all fine, right? It's that kind of initial part of transcription, right? So the catabolite activated protein helps RNA polymerase do that. And so if the catabolite activated protein isn't present because it doesn't have cyclic AMP bound to it, then more often than not, the RNA polymerase won't start transcription because it's either not found the promoter or it's fallen off of it, right? And so essentially what high cyclic AMP levels do is via the catabolite activated protein, it basically makes RNA polymerase more successful, right? And so there are more transcription starts per RNA polymerase kind of binding event, right? And so you get more mRNA, polycistronic mRNA. And the more mRNA you have, the more protein gets produced. And so the more stuff gets made, essentially. And so that's why it's a really cool fine-tuning mechanism. And that's why there's a kind of feedback loop going on, because the output of the things that the lac operon encodes protein-wise is glucose. And so once you get enough glucose, or you actually get onto glucose as a food source, right? Two different possibilities. You want to dial back gene expression, right? So in the lac operon, gene expression is highest when lactose is present, because that removes the LAC-I inhibitor or repressor, and glucose is low, because that means cyclic AMP is high, so you have more catabolite activated protein helping RNA polymerase do its job. So always think about, you know, what's the outcome? Like what does that organism need in terms of function, right? If it doesn't have any glucose and it doesn't have any lactose present, then you don't express the lac operon, right? Actually, it'd be an even worse thing to do if you think about it, because that takes energy. And if you don't have good food source around, the last thing you want to do is be wasting energy on stuff that doesn't benefit you, right? And similarly, you know, once glucose levels increase through either the action of the lac operon stuff, you know, beta-galactosidase and the like, or that happens to be, you know, you've just had a nice full carb diet, you know, Coke or something, and the bacteria in your gut is like, oh yeah, glucose. Sound like a dog rolling around in the grass. Then obviously you don't need lac operon expression, even if lactose is present, because you've got another food source. Yeah, so always think about what's the end result that is needed or desired. Cool question. Cynthia, you parked up yet?
I am, but you kind of answered the question I had.
Well, that's no good. Think of another one. Wait for you to park. Can't just like bow out like that.
The confusing part for me was that lactose. If that lactose is present, that's what the question that I had, but you cleared it up for me.
Well, if lactose is present, what does lactose do, Cynthia, in terms of the lac operon? In terms of transcription of the lac operon, let me be more specific.
Wouldn't it, if the okay, hold on.
So remember that the lac operon is inducible.
But it would continue transcription, right? Because.
But why? What does in terms of the control of the lac operon, what does lactose do? What's its role? Not in terms of making glucose, right? Metabolism wise, in terms of the control of transcription. What does lactose do?
Are you asking like that it encodes proteins? Part of it?
That's the lac operon does for sure, but the lactose, the actual metabolite, the disaccharide.
Oh, okay.
What does it do in terms of the control of transcription?
Are you saying would it stop or could basically like pause transcription or continue transcription?
Think about it. The lac operon is all about producing the stuff that metabolizes lactose, right? So you only want it to be expressed when lactose is present. Otherwise, you're wasting energy. Right? There's no point producing an enzyme to break down lactose if you don't have any lactose, right? So, one, the lac operon is off in the absence of lactose. 2, the lac operon is inducible, right? Think about what that means. It has to be told to be turned on. And 2, lactose is the inducer. So how does lactose induce expression of the lac operon?
Oh no. Okay, how does it induce?
Yeah. In other words, what does it bind to? What's stopping the lac operon from being expressed when lactose is absent? Right, because it's inducible, so it has to be induced to work. So something's stopping it from working normally. What is that something?
Oh my goodness, I'm looking into this paperwork.
It's a repressor protein. That's correct, Jacqueline. 100%. What's it called?
I thought it needed.
Say again, Cynthia.
I thought it would need the protein to continue. So the repressor protein stops, or it binds to the lac operon, or
Correct.
Oh,
and it
okay.
stops transcription because it's physically in between the promoter where the RNA polymerase binds and the structural genes, which are what, make the various enzymes.
Can you say that again? What she owed to?
Oh, I might need Cynthia's help with that. Cynthia, what did you owe to?
You cut off. I'm sorry. What?
Cynthia, what did you owe to?
What do you mean? What did I owe?
Oh, Kiana, yeah, you two need to have a conversation.
Oh, you were saying something. I guess you realized something, and you're like, oh.
Well, the thing is that I thought that okay, so according to this diagram, the repressor protein attaches to the lac operon.
Yeah, the lac operator.
Got it. Okay, so it's not the lactate. It needs lactase in order to continue transcription.
Ooh, just to be able to happen. But why? Okay. So you need lactose present for the lac operon to be transcribed. But why? What does lactose do?
It shuts off transcription?
Nope. That's what the repressor protein does. So what does lactose, the metabolite, do to the repressor protein? Remember, the lac operon is inducible, right? So it's off because of the presence of the repressor protein.
So it turns it on?
Yes, but how?
By binding to it?
Yay! Exactly.
My goodness.
Yay! That's worthy of a sip of coffee and maybe a bite of my cereal bar. So lactose is the inducer, right? In the lac operon. And it binds to, can anybody remember the name of the protein that lactose binds to, the repressor in the lac operon system? It's lac and then there's a letter after it.
Is it lac-I?
It is. Lac inhibitor is one way of remembering it. Good stuff. So no lac-I on lac-O, it gets a little confusing, I admit, equals RNA pol is able to transcribe the lac operon. Does that make a bit more sense now?
Yes, I'm glad I asked.
Exactly. All questions are good questions. So now you can see how essentially the thing that the lac operon utilizes, substrate, I guess, is the thing that turns it on, right? So the two things are tied really tightly together. Now, how about the trip operon? What kind of expression system is the trip operon? Is it repressible or is it inducible?
Repressible?
Correct. And so what does that mean in general terms?
Isn't it already on, so it has to be turned off?
Yeah, that's it exactly. And so again, think about like what's the outcome, right? It has to be repressed or turned off to you know, respond to levels, actually levels of what? What is the metabolite that is the co-repressor in the trip operon system? Presence of what turns off the trip operon?
Was it the CAMP one?
Nope, that's the fine tuning of the lac operon. So the trip operon is repressible, so it has to be turned off. It's always on. It's also a good example of a negative feedback loop. Right, so what thing does the trip operon produce? Well, the products of the trip operon. Trip is short for what? Give you a hint, it's an amino acid.
Tryptophan?
Tryptophan or something?
Yes, exactly, tryptophan. And so trip operon is repressible, it has to be turned off. It has a negative feedback loop, so the thing that it produces turns it off. So tryptophan is the thing that turns off the trip operon, but it can't do it on its own. What does tryptophan have to bind to for the trip operon to be turned off?
Is it tri-R?
Yeah, it's TRIP-R. And so what's the kind of formal name for TRIP-R? It's a repressor, but it's not like LAC-I. It's
aporepressor.
So what again?
An aporepressor.
Exactly. And what does that mean? What do we mean by an aporepressor? So basically, how is TRIP-R an apo? If I can spell that right. Sorry, should have been a question mark there. I mean, if any wags in the audience will go, well, they're spelled differently. That's one difference. Just before you think about saying that. So what's the functional difference between an aporepressor and a repressor?
Is it because the aporepressor is turning the operon off and the LAC-I repressor is turning it on?
Nope, they're both repressors. They both turn something off. Good guess though. Why is TRIP-R called an aporepressor? That's the key. Think about how the two different repressors work. So LAC-I is always bound to the lac operon operator until it's told to leave, right? And it's told to leave by what? Bit of a recap. What induces the LAC-I repressor to leave the DNA? Come on, it's up, further up in the chat. What induces LAC-I to leave the LAC operator?
Lactose?
Yeah, lactose. Cool. Okay. TRPR is different. Can TRPR alone, in the absence of tryptophan, bind to the TRP operator?
No.
Okay, why? What does it need?
A co-repressor?
Co-repressor, exactly. And what is the co-repressor for TRPR? Think about the negative feedback loop.
Is tryptophan the co-repressor?
That's it, exactly. So when tryptophan is not present, TRPR cannot turn off the TRP operon. That's the difference between an aporepressor and a repressor. A repressor can bind DNA as is. An aporepressor cannot. It has to have a co-repressor bound to it to kind of change shape to be able to bind onto DNA. Okay, so when you've got loads and loads of tryptophan around, are you going to get expression from the TRP operon?
Yes.
Think about what we mean by a negative feedback loop. Jesus is shaking his head there. Now he's like not moving his head at all. I can just see his eyes moving.
What was the question?
Sorry. If there is lots of tryptophan around, for whatever reason, will you get expression of the TRP operon?
No.
Correct. Why?
Because you need the TRPR.
Ooh, if there's lots of tryptophan around, there's always going to be TRPR around. That's the same for both the LAC operon and the TRP operon. So you have lots of aporepressor around. You also have lots of tryptophan, which is the co-repressor, right? So the two of those get together, bind to the TRP operon operator, no transcription. That makes sense? That's why TRP operon is repressible. It is normally on, but then it gets turned off. It gets repressed. Okay, does that make sense to everybody? Everybody all clear on that? Cynthia, how does that make sense?
Yes, I think actually going one by one, by breaking it down, you're able to see like the entire picture versus I look at the picture and I'm like, what is happening here? So thank you.
No worries. Always think about what needs to happen, right? If you don't have something, you need to make it. If you've got lots of something, you don't need to make it. If you've got something you prefer more than something else, then you're going to use that in preference to the other thing, essentially. Right? So think about always like, what's the ideal outcome? And how can we get to that? And really that works back to how this all works. And the trip operon is always on because you always need tryptophan, right? If you're a growing bacterium, you know, going to school, you know, hanging out with friends, that kind of stuff. You always need tryptophan because tryptophan is part of protein, so you need it for translation. However, you don't need like monster amounts of it, right? It's only one amino acid. So you need to dial it back when you've got enough. Lac operon is completely different because the lac operon is essentially lactose is an alternative food source. If you put a bowl of glucose and a bowl of lactose in front of a bacterium and ask it to choose, 100% of the time it's going to go to the glucose because it's a simpler sugar. It's a monosaccharide. It takes less effort to metabolize. So you only want to metabolize lactose if it's present and there's no glucose around, right? Because otherwise you just use the glucose. Okay, so next question, and this is in many ways the hardest part of this whole section, like all of it, including eukaryotic gene regulation. How do you attenuate expression of the trip operon? Say you have some tryptophan around, but not enough for TRPR to bind to the operator and shut things down completely. So say you've made some tryptophan and now you need to just dial back the output of the trip operon. How do you do that? Pardon me. How does attenuation work in the trip operon? Guarantee this is the subject that students struggle the most with because it is not easy to get.
Can you say that again, please? I'm sorry.
That's okay. How? So let's kind of, again, put up like the, I guess, the outcome, I guess, that we care about here. Say you have some tryptophan available, you know, enough to keep you going for a bit, but not so much that TRPR binds to the trip operator and shuts expression down. God dang it, completely. How is the trip? Actually spelling it trip, isn't that funny? Operon expression attenuated. If I can spell that. And attenuated means cut short. My time here has been attenuated, for example. So what's going on here? You can throw out any part of this. You don't have to describe it in complete detail. Okay. What's
involved? Does it have the leader region?
Leader region? Okay, that's cool. What does the leader region consist of? Oh, actually, where is the leader region? First off. Relative to the structural genes and stuff. Is it, where is it? Is it after the structural genes? Is it before the structural genes? Is it in the middle?
What slide are you looking at, Jesus?
I'm looking at my notes.
Oh.
Mm-hmm.
Now, let's see if I can find. This would be 17 and where is that? There we go. See it? Nader. Oh. Oh, out tabbing didn't work there. Give you a hint. It's the thing that's circled. So where's the leader region?
Like the 162 part?
Mm-hmm. Yeah, where is it relative to the structural genes? So TRPE, D, C, B, and A are structural genes. Those encode the proteins that make tryptophan. So we have the TRP promoter here. We have the TRP operator, right? So this is all about RNA polymerase and TRPR. This bit here. So if TRPR is bound with tryptophan to the operator, no expression happens. It's pretty tight. Okay, what happens if TRPR is not present? Do you get transcription or not? Don't forget,
No.
TRP operon is repressible, right? You have to turn it off. So if the thing that represses it is not present, it's going to be on, right? So nothing blocking the way here. We have RNA polymerase binds to the promoter here, TRPP. There's no TRPR here. We get transcription. What gets transcribed first?
TRPL?
Right, and L stands for leader region. Okay, so the leader region is transcribed first. It's part of the polycistronic mRNA, right, that will contain E, D, C, B, and A. So it's like part of the one big mRNA that contains all of this, but it's made first. Right? So that will make sense. Okay. So, more information. Tell me something more about the leader region. Anybody? What's special about it? Does it produce a functional protein or a structural protein? Yes or no?
You said it produces a structural protein?
No, does it? That's a question.
Oh.
Look down here, Jacqueline. Thank you for answering, obviously. We're still in the regulatory part of the operon. So this is mRNA, and it encodes protein, this leader region, but it's not a functional protein. So why produce a protein from the leader region? Right? Translation is expensive. Why would you make a protein from the leader region if you're not going to use it to do something? Structural, that is.
Wouldn't it still be produced because it needs something to bind to?
No.
Nope.
No, it's a short, say nonsense protein, but it's just a short polypeptide sequence that isn't used for anything. As soon as it's made, it just gets broken down and back into amino acids again. So why would you go to the expense of producing a short protein, however short, from the leader region if it's not going to be used to, metabolize something? What's the purpose of that short protein? Short polypeptide sequence, in fact. Why make it? It's part of the regulatory region of the trip operon. So it's got to be involved in regulation somehow, but how?
It controls whether transcription continues.
Exactly. That's a very good answer. Now, unfortunately, that leads to another question. How?
Because it's made up of 4 regions, and those regions can like make loops, and depending on which regions make a loop, then that affects whether transcription continues or not.
Okay, cool. So the leader region contains leader region mRNA. 4 different regions, 1, 2, 4, that can form how many different conformations, I guess?
Is it just 2?
Correct. Okay, so that's awesome. But how does the protein come into this? So you're 100% correct, right? We have 2 different conformations. Actually, it's worth putting one conformation equals transcription continues. Other conformation equals transcription stops. Attenuates is a fancy word for that. Because transcription has already begun, right? Because that we've made making the leader mRNA. What we're deciding is, does transcription continue into the structural genes, into TRPE? Right, that's what attenuation is all about. Now, the next question is, why are we making a protein? That's all 100% correct, but why make a protein? What's that got to do with this? What's the purpose of translation of the leader region? Hang on, let's dial things back up a little bit. So in terms of transcription and translation, what's one of the key characteristics of bacteria, prokaryotes? What can you say about transcription and translation in bacteria that you cannot say of eukaryotes?
Like they can happen at the same time?
Correct, exactly. They are coupled. So as soon as you get enough mRNA produced for the ribosome to bind to it, to the start codon, it will begin to be translated. Okay, so we're making mRNA. We're making the leader region mRNA. As soon as a bit of that is produced, we're going to start translating that leader region, okay, into that short, non-functional polypeptide. Why are we translating it? What does the ribosome do? What's the purpose of making that little protein? And linking transcription and translation are the key to understanding this attenuation process. Or I could put it differently. If you want to sense tryptophan, right? So remember in the lac operon, we sense glucose levels via an intermediate, right? Via cyclic AMP. We can sense tryptophan levels directly. So if tryptophan's amino acid and that's used to make proteins, how would you sense tryptophan levels? You'd want to see if there's enough around to make protein, right? And so what codons would you expect to find in the leader region if this is all about making tryptophan? So what amino acids need to be incorporated into that leader protein is another way of putting it. If its purpose is to sense tryptophan levels.
Wouldn't you need tryptophan or TRPP?
You would definitely need tryptophan, right? And so the trip operon, it's attenuated by using the translation of the leader region to detect whether or not enough tryptophan is present to translate that short leader region. And so if you're sensing tryptophan, then there's going to be a couple of trip codons present in the leader region, right? Okay, so leader region is the first bit of mRNA made, right? That mRNA has two different conformations, one which is pro-transcription, one which is anti-transcription, right? Remember, we haven't transcribed the structural genes yet, okay? Two, and that's why I asked, where is the leader region? It's before the structural genes, right? That's important. Three, we're going to be translating this leader region and using that translation process, right, making that polypeptide to sense how much tryptophan is present. Okay, that all makes sense? Those three things. Legion mRNA is produced. Leader region has two different conformations, one pro, one anti-transcription. And three, translation of the leader region is going to start before transcription of the structural genes. okay? And that leader region contains a couple of trip codons. Okay, now, how do we go from translation or not of the leader region to pro versus anti-transcription, those two different conformations? That's the question of the day.
Does it depend on how much tryptophan it senses?
That's exactly what it depends upon.
Okay.
But how is that not translated? How does that determine which conformation you have? There you go, this might help.
Is it because when the tryptophan is low, it doesn't bind, but when it's high, it does bind? No.
Yeah, I mean, that's right. When there's no tryptophan around, that protein is not going to get made. Whereas when there is tryptophan around, the protein will get made. But how does that turn into essentially these two different possibilities? Right, here's one conformation where you have region 2 and 3 bound. That is pro-transcription. Here's a different conformation where you have regions 3 and 4 bound. That is anti-transcription. How do we basically choose between these two possibilities?
Does it depend on whether region 2 is available?
It does, 100%. What determines whether or not region 2 is available?
Isn't it like the ribosome that's coding for it?
Correct. 100% correct. So we're getting there. We're getting to the point, I guess. And so, big important question, where are the trip codons in the leader region?
In between 1 and 2?
Well, actually, they're in region 1. Right, so pretty much the same thing. So what will happen if there is no tryptophan present in terms of translation and the ribosome?
The ribosome is stalled?
Yes, where?
On region 1.
Okay, so if it's stored on region 1, what's going to happen in terms of these two conformations? Right, so region 1 is not available. Region 2 is. And so what region gets transcribed after region 2?
Region 3.
Okay, so if you have region 2 and 3 around, you're not going to have region 4 because it hasn't been made yet. So if you have region 2 and 3 available, which of these two conformations are you going to get?
The pro-transcription.
Exactly. So low tryptophan levels equals more transcription equals making more tryptophan. How cool is that? and it's sensed directly. Go on, Kiana.
Sorry. So does high tryptophan levels mean, because you just said that low tryptophan levels means more tryptophan is made, right?
Mm-hmm.
So then with high levels, less tryptophan is made?
Right, so good point. Now we get transcription of the structural genes. We get tryptophan being made. So tryptophan levels start increasing again. So we still have transcription occurring of the leader region and then translation occurring of the leader region. Now we have tryptophan around, enough at least, for translation of the leader region. So what's going to happen now in terms of translation of the leader region? Is it going to pause or stall on region 1? If there is.
No.
Exactly. Because there's tryptophan around, right? So if it doesn't stall on region 1, where's it going to go? What's after region 1?
Region 2 and 3 will bind.
So hang on a second. So remember, transcription and translation are occurring at roughly the same speed, like that, along. Right. So the only reason why 2 and 3 will bind is because transcription continues along like so, and the ribosome pauses. Like so. So region 2 and then region 3 is made, and then they form that loop which prevents 3 and 4 from pairing. Okay, so essentially, if tryptophan is not present, you get a decoupling of transcription, which continues, and translation, which stops, or stalls is a better word. Now, if you have enough tryptophan present, you still have those two things coupled together, right? So the ribosome travels in to region 2, right? So is region 2 available to pair with region 3 when region 3 is made?
No.
Okay. So translation continues and transcription continues. So if region 2 isn't available, what's going to happen when region 4 is transcribed?
It's going to bind with 3.
That's it, exactly. And if region 3 and 4 bind, they do so inside the RNA polymerase because of this U region, which slows down the RNA polymerase. If you have a hairpin loop form inside the RNA polymerase, what happens? What happens to the RNA polymerase? Does it go, ooh, that tickles, and just continues? Or
it terminates transcription?
Yeah, it terminates transcription. See, it's really, really neat how this works. So now if we have enough tryptophan that translation continues, but not enough for TRPR to bind, right? So we're kind of somewhere in between, in the middle. Right, so we want to maintain tryptophan levels around where we want them, right? Like homeostasis, a little bit of bouncing around. So now we have enough tryptophan for translation to continue, and it blocks region 2 from forming a loop with region 3. Now the RNA polymerase kind of makes 4, pauses for a bit, and then we have the termination loop in between, which terminates transcription. So we make a little bit of mRNA, the leader region. We make a little bit of protein. essentially regions 1 and 2 are translated, but that's it. So there's a little bit of cost, but there's not the expense of the whole tripoperon being transcribed and translated. You see, it's not trivial. It's actually, there's an awful lot going on here. And it's basically the interplay between translation and transcription, and particularly RNA structure and transcription. Questions? Did that make sense for everybody, or is there stuff that I didn't quite get right?
It makes sense.
Cool, because I'm going to ask you a bazillion questions on this stuff. In lots of cruel and unusual ways. Oh, perhaps not cruel or unusual, but I will be asking you lots of questions. Cool. I'm kind of glad I screwed up the whole exam date thing. This has been really good. Obviously not glad because that makes me look like a bit of an idiot, but we get to have this instead. All right, questions. Anybody got questions? And don't forget too, in eukaryote gene regulation, there is a whole wealth of stuff, fertile ground for exam questions. Lots of details, lots of this versus that. Pardon me. All right, I haven't heard from yet. There you go. Mika, you've been very quiet. Got any questions? Questions. Oh, that's cool. Oh, fun fact. I got to touch a giraffe over Thanksgiving. Super cool. I was very excited by that. Small tail.
Was it good?
Yeah, I only got to touch its nose, just like with a finger, because it was leaning over the fence. But it was closest I've ever been to a giraffe, and I've seen lots of giraffes. It was very, very cool. We stayed at a campsite that had a near Ubaldi, which had like a safari kind of hayride thing. You kind of got dragged around this really huge field with lots of really exotic animals. Springbok and camels and all kinds of stuff. Ostriches, emus. The ostrich was pretty vicious, so I stayed away from that. Actually bit my daughter's hand, which was, probably shouldn't say it was funny, but I wasn't super surprised. All right, everybody. Well, we're pretty much near the end of the session. This will go up on YouTube, Blackboard in a bit, probably after lab, I would guess, maybe this afternoon. So, as always, study hard and studying kind of try and think of different ways in which you can study, right? To make this stuff take and sink in. Because as you've seen from today, you know, there are different ways that you can approach this. And some of them work better for you than others. And that's going to be different from person to person. Right, so don't just read over the notes and hope it sinks in, because if it didn't the first time, there's a good chance it's not going to the second time or the third or the fourth, right? You got to understand this stuff to be able to remember it well. All right, and if you have any questions whatsoever, just ask, right? I will respond by email. I've got a whole bunch of lab reports to go through. So those are next on my list of things to do. But I'm always available for either answering questions or even meeting up via Zoom if you need to. So if you have a whole bunch of questions that you can't answer yourself between now and the exam, just hit me up and I'll help you answer them. Cool. And don't forget the SRIs, a little bit of extra credit there for you. Cool. All right, everybody. See you all soon.